Okay, I think we are ready to go. So, chapter 6, I would like to think is all about viruses, but there's a little bit more. Uh, the bulk of it is going to be virus, and we're going to think about bacterial viruses, bacteriophages, and then other viruses, plant and animal. We'll talk a little bit about some plant stuff right at the start. And then we want to think about some other pathogens, stuff like prions. I like to think of that as mad cow, but it's more than just mad cow. So we'll talk a little bit about that and some viroids, a little bit different as well, toward the tail end of this. So again, this is introductory stuff. As we start moving through the actual disease, we will hit a handful of viral infections and provide some more detail. So whenever I start this and I see the Ebola up there, it is a type of hemorrhagic viral infection. So bleeding, there's all sorts of descriptions about Ebola. Hot zone does a pretty good job describing these issues, a little embellished, but impressive and disturbing. There are lots of different hemorrhagic viruses. Ebola is one that is pretty famous, I suppose we could say. But there are a few others, and we'll touch on some of them as well, like Marburg and Lassa. If you are interested in things like this, there is a book called The Coming Plague. Uh, I want to say Laurie Garrett is the author. I could be wrong with that. Uh, very interesting accounts looking at a handful of doctors studying and traveling to Africa, studying all sorts of different hemorrhagic fevers and thoughts about some of these, blowing up and causing, pandemic sorts of situations, which I think is truly pretty terrifying. And pretty thankful that our pandemic was coronavirus and not Ebola virus. Would have been a very different picture. All right, I'm not going to go into details with that. Not yet. Yes, sir.
How contagious was the Ebola virus?
How contagious is Ebola virus? Super contagious. Coming in. So basically, infections with that, you have all sorts of issues with your blood bleeding, pretty much from any and every orifice. Coming in contact with any of that from a patient ultimately sets the stage for infection. So super contagious, and mortality rates very high, right? Few able to actually survive that, but definitely, some terrifying stuff. When you think about that, nightmare worthy, I think so. There's many things in here that potentially could be some nightmare worthy situations. So I told you we'd see some plant stuff. Tobacco mosaic virus. This virus has been a virus that has been studied for a super long time. We talk about TMV, or tobacco mosaic virus, in biology texts when researchers were setting out to understand the genetic material. And the questions were, is the genetic material, what is this information for life? Is it nucleic acids or is it proteins? Both held potentials that went to, two schools of thought, thinking these are truly the molecules that confer the instructions for life. there's a lot of variation when you start thinking about proteins, and speculation was that seems to be fitting. There's variation in nucleic acids as well. So lots of experiments set up to truly try to figure out what's happening. They used different types of enzymes to degrade things. DNase breaks down DNA, RNase breaks down RNA, and proteases break down proteins. So taking samples and exposing them to their specific enzymes allowed them to truly get to the heart of the matter and recognize DNA truly is the information containing molecule. Tobacco mosaic virus was utilized in that process to truly kind of solidify those ideas. And viruses, as we've already kind of touched on the littlest bit, super small, considerably smaller than our bacterial friends. The bacteria are like giants to a virus, and we are like giants to a bacterium, thinking our own individual cells, not just us and a little virus. Then, I mean, we're monsters, right? So filterable viruses, you can use all sorts of different filtration techniques, trapping specific things. I believe we'll see that here in just a second. And the virus is capable of moving through a filter that traps pretty much everything else. All of the bacteria gets left behind, and the virus can go through, still harboring the potentials for infection, and then through digestions with those enzymes, truly identifying not the protein, but the DNA as the culprit. So this image next to it, figure B, shows a handful of tobacco leaves that are infected with the mosaic virus and the issues that you can see on the crop. We will think more about a little of that as we progress. This image is of a bacteriophage, so a virus that infects bacteria. And as far, there are a lot of different morphologies for virus. And with some of these bacteriophage, different potentials, these we call some like T4 bacteriophages. And they're some of my favorites. I think they look awesome. This, as we are looking at this electron micrograph, this big area, darker gray, this is a bacterial cell, maybe E. coli, all right? And the bacteriophage in the process of attaching and infecting that cell. We'll go through the process of that stuff here in just a second, all right? So I like to kind of imagine this virus, as I look at it, it kind of looks like some sort of spacecraft, some sort of alien, potentially, spacecraft landing in a new area, a new habitat, a new planet, a bacterium. And what do aliens do? They probe, right? That's what ultimately starts happening. They pierce into this cell, and then they deposit their genetic information. Pretty impressive. Once a cell, doesn't matter if we're talking bacteriophage or our own bodies, once a cell becomes infected with a virus, that cell pretty much stops being a normal cell and shifts over to a virus producing factory. That's really its job. And how it goes about pumping out virus, if you will, truly depends on some of the cycle of that virus. Sometimes they replicate and explode out of it, and other times they lay back and incorporate themselves into the host chromosome, replicating with those cells, and then later on emerging and releasing virus. And in that instance, now if we're thinking bacteria, you could have countless bacterial cells that have replicated. I mean, you could get millions of cells in a day if we're thinking a culture of bacteria. And if all of these cells, if the first one was infected, all of them have it, and then they can potentially emerge. Yes.
Once the virus shift from that dormant phase to like an active phase.
Ooh, that's a great question. Shifting from the dormant to the active. So we're going to get to a slide in a second that shows a little of this. That bit, we call that lysogeny, when the virus gets in and takes residence in the chromosome. All right. And then after rounds of replication, it can go into the lytic cycle, which is just reproducing and rupturing out. All right. So there's a lot of different things that could potentially stimulate to those cells problems, right? It could be environmental shifts, so nutrient depletion, waste accumulation, something that potentially the bacterial cell may start to sense poor issues in its habitat, might be dying, right? The virus ultimately gets activated in those circumstances, and there's numerous molecular cues like that that the cell ultimately is starting to recognize or produce, and that's what stimulates a lot of the lysogeny to lytic transition. Pretty cool, all right? And also a little bit terrifying when we think about that. This image, I think I showed you a different image before because I told you I wasn't super happy with the one that the text provides. This is from the text. Now, why I'm not happy with it is because over here where we see what the light microscope can see, we're bumping it right over to show you, oh, well, we could see smallpox and flu viruses with a compound microscope. That's not true. That's a stinking lie, all right? The light microscope should be ending right over here at about a micron. Bacteria, we can visualize. You've all seen stuff. You did a little bit of gram staining, super small, right? Viruses, there's no chance you're going to be able to see a virus with a light microscope, all right? So yes, the electron microscope can bump over here. We can see bacteria with the electron microscope. You saw a fraction of the bacteria when we were seeing that phage attached to the surface, all right? So considerably smaller, all right? And something that truly shouldn't be picked up with a light microscope. So depending on the virus, there are differences as far as how they emerge out of those cells, all right? In some instances, the virus replicates and causes that cell to lyse, to basically break apart, and you're left with more virus that emerge out of that cell. Viruses that do that inside of the cell, we call those naked viruses. They just release out. Other viruses inside of the cell start replicating and then they bleb off. They don't cause the cell to get destroyed. They get released out, just like exocytosis, where you're taking your waste out in a vesicle. That virus, as it's leaving the host cell, coats itself in the plasma membrane of that cell. Now it's got a shield around it, all right? Human stuff, human membrane circling around that virus. We call those enveloped, all right? So bringing extra stuff and extra coating. So adenovirus is a naked HIV, an enveloped virus. I'm not going to spout off details about those right now, even though I want to, all right? We'll touch more on it later, all right? So as far as morphologies go, this is also varied as far as how they may appear, all right? The capsids are the protein shells, if you will, that harbor the nucleic acid inside, all right? And these capsids could be naked or enveloped, all right? As far as their shapes go, it could be like a big long tube, cylindrical or helical, all right, with proteins circling around it and then the nucleic acid inside, all right. They could be like that T4 that we just showed, kind of crazy shaped, all right. Icosahedral, as we see here, rhinovirus, one of the common cold culprits, looks this way, all right, icosahedral. There's others like those phages where you can see the tail pins and all of that stuff as they're making contact. We'll show some anatomy of that in a second, all right? And then there's others that are kind of more irregular or complex shape, variola, as we see up here. Chicken pox, smallpox, they're variolas. Variola zoster is our chicken pox. And as we progress along, it's within the herpes group of viruses, all sorts of different herpes. All right. So the first bit, and we're looking at bacteriophage, all right, but there can be some similar ideas thinking animal virus as far as this process of infecting, maturing, and being released out. All right. So we start off up at the top, first bit with the attachment phase. So our oval here is a bacterium, and then we've got our cute little bacteriophage up at the top. All right. So the bacteriophage, there are all sorts of variations with this. And I know we've mentioned a little of this before, but specificity, all right? The little feet on this virus make recognition with a host cell. And if it's the proper host, their proteins, their little feet, as they make recognition with that host cell, link up with specific receptors and basically allow that virus to know I'm on the right thing and it can infect it. And if it comes in contact with a cell that doesn't have those proteins, that's not the right thing. All right. So a lot of specificity, and that holds true when we think about virus infecting us. Pretty much specific with things or individuals that could be potentially plagued by it. Some of them are a little more generalists. Rabies pops into my mind when I think about that. If you're a mammal, you're fair game, all right? But other things, definitely more specific. So bacteriophages oftentimes harbor, oops, going the wrong way, tremendous specificity, and that sets the stage for maybe possibilities of using a bacteriophage to target and treat some sort of infectious disease because they only want to get that one type. There are definitely opportunities for some of them to have a little bit of wiggle room, and some that are maybe close cousins. There might be a little bit of broader host range that we may be able to witness. Depends. All right. So we make contact with the proper host. All right. And that's when the attachment phase happens. Everything's perfect. We lock up on there. And as it attaches to the surface of that cell, then the probing happens. All right. The penetration phase, this phage, we've got our capsid up here at the top. And then this sheath right here, this rod is, I kind of imagine it's sort of like a hypodermic needle. All right. There are some pins, and we will see a blow up image of a phage in a second, but there are some little pins like the needle on the end of this sheath. And if we have made contact with the proper cell and able to attach, we penetrate in, pushing those pins through the cell wall of the bacterium. And like a hypodermic needle, as we penetrate in, the DNA is shot into the cell or inoculated. All right. So phage DNA, host DNA, and like I said, typically once this has happened, you're no longer behaving like a normal cell. All right. Host chromosome winds up getting impacted, degraded, maybe taken out of commission. We utilize this host's machinery, free floating ribosomes, to make copies of the phage DNA. And we use not the ribosomes to make copies of the DNA, but polymerases, sorry, ribosomes to make the proteins, the capsid, the shell material for that virus. So we've got free floating viral DNA, and we've got capsids that are being generated. All right. This is the synthesis or biosynthesis phase where it's making new progeny. All right. The shells and the nucleic acids needed. All right. After that, we get the maturation phase. This is where they spontaneously assemble, and the nucleic acid gets packaged in the capsid. It packages it all up and puts it together around it. And now we're ready to go, locked and loaded. All right. The phage gets assembled in the maturation phase, and then lysis is the end result. And the bacterial cell, once these guys are all mature, ruptures, releasing out phage. All right. And as we see in this image, we get a single phage infecting a cell. And at the end, we'll get a half a dozen or so. All right. It's not like this one bacterium's pumping out millions of virus, a handful, maybe a few handfuls, right? Half a dozen, maybe a bit more. All right. Those bump into new cells and the infection keeps going. All right. So this lytic, all right, and we call these virulent phages. All right. The virulent phage infects, replicates, and destroys that cell. And typically this could happen in half a day, maybe a bit less. All right. Pretty quick to start this process of replication. All right. The temperate phages, not virulent, temperate phages start off with the lysogenic cycle and then go lytic. All right. So this gives that phage an opportunity to persist. It gives it an opportunity to replicate along with the bacterium because it's incorporated into its chromosome, and then later on, releasing it out. So now if we flip it back thinking lytic, and we get a single virus infecting a single cell, and we pump out 6 virus progeny after that, virions, after we have done that, we've only made 6. But if we go this route with lysogeny and a single virus infects a cell, now after a day's time, we've got 1 million or more bacterial cells in this culture, all of them infected with it. And then they hit the lytic cycle. And instead of releasing 6 out, now we've got 1 million bacteria and each of those releasing 6. So now we got 6 million virus instead of just 6. That's a brilliant strategy, right? Now we've released boatloads of virus, right? So incorporating in, it's the same thing. It attaches, it penetrates in, but then it incorporates into the genome. When you get bacteriophage DNA incorporating into the host bacterial chromosome, we call this prophage DNA. All right. When this happens in our own cells, we don't call it a prophage because phages infect bacteria. We call it a provirus. Proviruses, people, prophages, bacteria. All right. Same idea, but different name. All right. So After it gets in there, it replicates, then some sort of stressors, and it pops out of the chromosome, and we move into the lytic cycle. We replicate, we mature, and we rupture out. All right. Same deal, right? With the single cell pumping out a handful. All right. So kind of a brilliant strategy. All right. When we think about phage therapy, using a bacteriophage to treat some sort of infection, you technically don't want it to be these guys. All right. You would like your virus to find that host cell and to destroy it as quickly as possible. You don't want them to find it, to get in there and hang out, and we're just going to live in these cells while we're causing disease in the host, ample nutrients, all fine and good. The bacteria is replicating like gangbusters, right? We're just pumping out cells because conditions are super favorable. That phage is never even going to have a chance to do its job because the bacteria are in optimal situations, right? Maybe long after you're super ill, it finally does this. Then it's too little, too late, right? Virulent phages are what we would truly like to focus on if we're thinking about therapeutic agents, all right? Not that there isn't interesting details about lysogenic phage, but treatment-wise, a little bit different, all right? When it comes to molecular biology and we think about genetically manipulating things, oftentimes we use plasmids from a bacterium and we put a gene of interest onto that plasmid and pop it back into a bacterium. There are other techniques where we can use virus to infect and incorporate different genes into a host cell. That can be utilized as well. And this mechanism is ultimately what kind of lent to the ability to do that, recognizing lysogeny and incorporating in. In this case, what we're looking at is specialized transduction. So 3 ways that bacteria can manipulate things. We think about conjugation, sharing plasmids. Transformation is just pulling in DNA through the environment, and transduction is virus that's incorporating stuff. We'll touch on those a bit later, all right? So the specialized transduction, we've got our virus up here, it attaches, penetrates, we've incorporated this nucleic acid into the host chromosome, so integration, all right? And then this is the specialized bit, all right? Sometimes when that phage DNA is excised, we excise a little bit extra, all right? Now we've got a chunk of host chromosome along with viral chromosome, all right? And there is the potential for these two to get packaged up into a new phage and to be infected into a new cell. And that's what we've got going on right here. Blue and pink, both coming into the mix. Now, this new bacterial cell, not only is it infected with viral, it's also infected with host chromosome. And if we've got a virus that's got a little more broad range, we now target a cousin instead of the same, right? Now we give that cousin somebody else's DNA as well, giving it different potentials, right? When we think about using transduction in genetic engineering, we only allow the host, the chunk of DNA we want, we allow that in there, and the rest of the viral stuff not. So now we know this virus is only infecting with what we would want, right? In this instance, this is just a normal circumstance where some virus pick up some extra stuff, lysogenic virus, pick up some extra stuff, all right? And now bring it in to something new, all right? And we call that specialized transduction because it's bringing extra stuff. And now we've got a recombinant, all right? Somebody that's genetically different than how we started, all right? So I said similar ideas when we think bacteria versus animal virus. So this is showing the cycle of that. We start off up here at the top with the attachment. So this is flu virus, influenza virus. It's making contact with a host cell, all right? Epithelial cells within the mucous membranes. So it attaches to these target cells as the spike proteins on the viral capsid recognize host cells if it's the right cell, epithelial cell, they're brought in, all right? And that's what we're seeing right here. So the penetration bit, this is basically like endocytized. So we make contact with the host membrane, engulf it, and pop it in. So as we look at this, you can see our virus has a membrane encasing it, all right? So it's brought to the inside. Once it does that, then it hatches out, if you will. It uncoats, releasing its contents to the inside, and we move into the synthesis phase. In the case of influenza, I better put the brakes on real quick. I need to back it up. Viruses are a bit different. I mean, we don't call them alive in the first place. Viruses are different because what they bring to the mix, their nucleic acids can be very different, all right? A virus either is a DNA virus or an RNA virus. They never have both molecules. It's one or the other, all right? And then depending on what they bring into the mix, the cellular machinery of that host they've infected is going to work on that, all right? So it just depends, all right? And then to even make it more complex, the nucleic acids, doesn't matter, DNA or RNA, they may be single or double-stranded molecules, different, all right? Viruses harbor all sorts of kind of crazy ideas, and it makes me think back to early life on Earth. There's ideas of, the first cells with this RNA hypothesis that really that was the genetic information. And then later on, as we think about potentials for that information to maybe be targeted or issues with it, folding back or complementary pairing upon itself lent to a double-stranded molecule, DNA, all right? Different, all right? And that's just a small little tip of the iceberg with the RNA hypothesis, all right? But visualizing differences in the viruses and seeing a virus that only harbors our RNA molecules kind of goes to support that hypothesis that maybe truly the first forms of life really were RNA molecules for their nucleic acids, all right? So we get in there, we start synthesizing the RNA from our flu virus, all right, gets into the nucleus. Within the nucleus, this is where it starts getting replicated with RNA polymerases. It starts assembling in our assembly phase the capsid material for the influenza. So all of that starts getting assembled along with copies of the RNA blueprint. All of that gets packaged up. So the maturation phase is we're putting it all together and then released out. All right. So as this happens, and the cell is pumping them out, we see our flu virus released out and ready to link back up to a new cell. All right. So the RNA stuff. But like I said, there truly is all sorts of variation. So in this case, we're looking at our single-stranded RNA molecules, and they may be negative or positive sense. And when we think about transcription and translation, as we make a copy of that instruction from the DNA, we're reading our DNA, we pump out our messenger RNA, and as we are doing this, the orientation 3 prime, 5 prime, right? As we are building these molecules, it truly depends on the orientation of that RNA molecule that infected. All right. What is it? All right. If it's positive sense, you're good to go. That messenger RNA is written in the correct order. All right. And ribosomes can snag onto it and we can start pumping out proteins. But if you're in the opposite direction, going backward, right? We don't read it that way. So we have to convert the negative to a positive strand. There are some enzymes that help with the conversion from a negative to a positive. And then we're able to work upon it. All right. Impressive, I think. All right. So this image showing HIV and the cycle of HIV later on, I don't think it's in this chapter. I think it's later, pretty sure. Maybe I'm wrong. Maybe it'll probably be the next slide. I'm like, dang it. As we look through this process of the virus infecting, there are numerous steps in this cycle that we think about as potentials for targeting these processes for treatment. All right. And oftentimes cocktail approaches where you try to target multiple angles of the viral cycle. All right. So this big blue guy right here is the cell that's going to be getting infected. HIV infects T cells, T helper cells specifically. All right. So they need to be in your body. All right. We think of blood-borne situations that allow access to these cells in the first place. Coming in contact with it and not actually getting it through the skin is not going to be a situation that's infectious. All right. So not that you should be touching HIV in the first place. So HIV is an enveloped virus. It is an RNA virus, all right? So it's got its spike proteins that recognize specific proteins on the host cell. These proteins we call CD4 cells. CD4 cells are T helper cells, all right? These CD4 proteins allow the HIV virus to recognize it, and then we are able to be brought in. All right. So first off, we recognize, all right, and then we're allowed to penetrate in. So the HIV RNA and some ingredients that it needs to make everything happen. It's not just RNA, there's some enzymes that it's bringing into the mix as well. The host cell is going to pump that out for it, and it's going to package it all up and go about its business. All right. So it brings in its nucleic acid, RNA. It brings in an enzyme called reverse transcriptase, integrase, and a handful of other proteins that help to get it where it needs to go and do its job. All right. Reverse transcriptase is an enzyme that becomes super important when we think about genetic engineering and the ability to manipulate stuff. We'll hit that stuff a little bit later. All right. So as this happens, viral DNA gets produced by that enzyme reverse transcriptase. It reads the RNA and makes a double-stranded copy of DNA. All right. So as that happens, now we've got DNA, we've got the instructions, and integrase, another enzyme, helps to facilitate the movement of that HIV DNA now into the nucleus of that helper cell. It incorporates into the chromosome. And then now we've got DNA that can be read, and we can make RNA. We're making the HIV blueprint. All right. So we build that. We read other instructions within that that say, oh, why don't you build me some reverse transcriptase and integrase? You build all of those things, packaging it up, blubbing out, and the cycle can continue. All right. So we will hit more of that as we progress through. All right. So we showed varicella, varicella zoster, chickenpox. Was it varicella? Maybe it was. I can't remember what it was. I said it was chickenpox. Now I'm like, did it say varicella? I could be wrong. So chickenpox, it's enveloped, icosahedral, and this next bit isn't chickenpox. It's secondary. So shingles happens in individuals that had chickenpox, typically when they were kids, and then the virus can go dormant on some nerve cells and potentially later on reemerge. It's kind of like the lysogeny, where it just hangs back. All right. If you have been vaccinated against chickenpox, technically should be good to go. So we think. All right. There are also vaccines as you start to get older. I want to say 60, might be a little bit earlier than that, where you can get a vaccine against shingles to help prevent it from reactivating. All right. So as far as the virus goes, this is like a growth curve or a multiplication curve of the virus. All right. So we've got the number of virions, viral particles, on the y-axis and a time frame. It's all arbitrary down here. We're not giving any numbers, but again, a single cell pumping out a handful, right? So we start off here with a handful of these virus. They get inoculated into a culture, maybe thinking bacteriophage stuff. So they get access to some bacteria. They bind. We see their numbers decreasing at this point because they're all hidden away inside of those bacterial cells. You don't see them now. So this eclipse period is where the virions are getting into those cells, hiding away. All right. And as they go through and start the assembly process, the biosynthesis and assembly, once they get all packaged up and start getting released, that's the burst. And it's like, exponential growth when we're thinking about a growth curve, where you see a dramatic spike in the amount of virus, in this case, that are being released out. And you get to a threshold up here, which is like back to that initial image, 6 virus per bacteria or whatever it is. That tells you the burst size. And you can grow them with bacteria and ultimately start to calculate this if you know some of the details about what you were working with in the first place. All right. So we've shown some biosafety force stuff. Working with Ebola definitely warrants this sort of laboratory situation. Only a few labs in the country to truly have the capabilities of working with BSL level 4 pathogens. So ventilation, positive pressure suits, all sorts of mechanisms to help ensure the safety of the personnel and the environment as well, to make sure that these things aren't being released to the outside. So this talks about some of the potentials for filters. Not the same filter that they were thinking about with the tobacco mosaic virus, but same sort of an idea. These are membrane filters, so it's got kind of like a filter paper sort of disc on the inside of this red plastic job, a syringe up here at the top. And what we're doing is filtering with different pore-sized filters, all right? So we've got a handful of different types of cells in this syringe, all right? The green guys are the virus. The red guys are blood cells, we'll imagine, eukaryotic cells, and the blue guys are bacteria, all right? So eukaryotic cells bigger, bacteria bigger than virus, and the smallest one's the virus. All right. So at a pore size of 5 microns, bacteria, typically about a micron or so, they're able to pass through a 5 micron filter, but the eukaryotic cells aren't. All right. So they get trapped, but the bacteria and virus go through. You get a pore size considerably smaller, 2 microns or 200 nanometers, bacteria are too big and they get trapped. And what's left behind are the virus. So filterable situation, all right? And we use these types of filters in our bacteriophage lab to try to purify out the virus from a soil sample that's chock full of bacteria. We don't want any of the bacteria. We just want the virus. All right. And then we go through and try to cultivate it. So this is like that with the phage lab, where you've got a known source of bacteria and you mix it with some potential virus. And if the virus is there and able to infect the cells, this is a lawn of growth. So it's all cloudy because you've got bacteria growing all over it. And every little circle in here or divot, we call these plaques, it's the result of a phage that infected the bacteria and then killed them. So no bacteria is growing there. They're all dead. And right here is a bunch of virus, all right, that's basically been released out. So this is a dilution where we've got high concentration of virus, little lower, and then even more dilute. And then you can start calculating about how much you've got in your vial if you do some of these dilutions and get something that you can count and then something that's too numerous. When we think about animal stuff, we grow them in cell cultures with human or other animal cells that have media that allows those cells to stay viable. And then we can look at these under like a dissecting scope and see those animal cells and the potential for them to have different morphological changes as the result of the infection, cytopathological effects, changes in the surfaces of the membrane of those cells that can indicate viral infection, all sorts of different things. All right. Sometimes we use, instead of, cells in a culture, we may use animals. So embryonated eggs, chicken eggs are a good one, where we could potentially use this to cultivate virus. Flu vaccines typically are produced in chicken eggs, although they are moving to some other techniques. This is still the common mechanism. Multiple ways that you could inoculate and in turn then harvest out and synthesize specific fragments of that virus that could stimulate an immune response, those spike proteins, all right? So this is some cell culture stuff. Primary cell culture and a continuous. Depends on the types of cells that you're working with in the lab. Primary cells are just normal cells, all right? They grow, and then once they come in contact with their neighbors, they stop growing by contact inhibition, proteins on their surface, all right? And you have to repeatedly transfer by taking some of these cells to a new flask with media, and they keep growing. But the continuous line, these come from cancerous cells that have lost contact inhibition. They don't know how to die, all right? And in turn, you can keep them propagating with minimal needs for transferring to new media because they just keep going, all right? And the HeLa cells, Henrietta Lacks, some of the main continuous cell lines that are used in research labs, I would say globally, all over. And there's some disturbing stories about her and all sorts of issues about that. I'm not going to get bogged down. I just want to say thank you to her. And I'll leave it at that. All right. She was kind of ripped off. All right. Okay. So few differences. I know I'm almost out of time, so I'm going to skip over that. There are some potentials. This is some immunology stuff to look at possibilities for viral infection. Normal stuff up here at the top, just red cells, no reaction, little button. All right. A virus in red cells. In this case, they want to target the red cells. They link up and it causes clumping, hemagglutination. All right. And then to test if the patient has antibodies against this virus, we could take serum from that patient, mix it with the red cells and with the virus. And if the patient truly does have antibodies, they link up to the virus and you don't get any agglutination. It looks just like the top. That tells you the patient's serum sample has stuff in it to target that virus. All right. Doesn't necessarily mean you are currently infected, but it could mean you've produced antibody. You maybe were exposed or you've been vaccinated against it and you've got antibodies. And the agglutination test allows you to detect that. This is an enzyme-linked immunosorbent assay, antigens and antibodies interacting together to give you those potentials, and another mechanism to pick that up. All right. Viroids, these bit different than our virus, but still within this category. All right. These are potatoes that have been infected with potato spindle tuber viroid. And this happens in the springtime with the seed potatoes. We are in Idaho. Hopefully, you know a little bit about potatoes. Seed potatoes, typically much smaller than the potato you think about buying in the store. They get cut in half or maybe quarters, and then those pieces get planted in the ground and you grow up a crop of potatoes. If those potatoes, if one of them was bad and it gets cut with the knife, every other potato that gets cut with that same knife ultimately becomes infected. Now you've got a whole field full of potatoes that look like this. All right. Not something that makes them not able to be eaten, but I'm pretty sure if you were going to the supermarket and see a bunch of potatoes that look like this, you would pass them over for a better looking potato. All right. Potato flakes, French fries. That's where we think about some of these things. All right. Prions, real quick. Let me just give you this. There's all sorts of potentials with prions, different animals, all sorts of different things. All right. Prions are normal proteins in your brain, endogenous prion protein, the green guys, the good ones. All right. They may, through just mutation events, could spontaneously form into this catalytic protein, the red guys able to do something. All right. Conversion of the mutant, right? So once this happens, the mutant can interact with the green guys, causing them to go red, right? So all of that can happen, or consumption of neural tissue that had this stuff going on, all right? Inoculation, all right? And that, all of these different instances that can lend to this. So it causes some spongiform encephalopathies. It causes sort of a Swiss cheese appearance within the brain. Lots of different animals as potential culprits. Humans can get infected. We think some spontaneous stuff. Creutzfeldt-Jakob disease is that, but the consumption of brain for a tribe in New Guinea as part of their death rituals would consume their dead. And if they died from this, those that ate the brain tissue ultimately decades later typically wind up with the same thing. All right. So a lot of issues, and we call that kuru with those individuals, but it's same sort of an idea. In this case, inoculation to get it. Yes.
Found in like nervous tissue.
Yes, nervous. So that's the idea, nervous tissue. And that Creutzfeldt-Jakob versus the normal, and then the sections like this. Kuru would look very similar, but this, like a spontaneous situation, Kuru was through ingestion, same sort of an idea. Coming in contact with animals or eating brain tissue could set the stage for that. Lots of different opportunities. And with livestock, there's different names for some of these. Scrapie, we can see in sheep, chronic wasting, which is in like deer and elk populations. That one's a little bit different. And transmission with it is through saliva, which is concerning and disturbing. All right, I apologize that I went a bit long, but thank you for bearing with me. Test opens in mere minutes, and closes Monday at 5. All right, good day, farewell.